Good evening. Let me start by introducing myself, Mosa Moshabela, the new Vice-Chancellor here at the University of Cape Town. I'd like to welcome all of you to the session. It's a very, very special session today. I also know that we've gone live. There's quite a number of people that have joined us online as well. And we're very excited that we have a great audience for the lecture today. Let me specifically welcome all the executives that are here, DVCs, and I know that the former interim vice-chancellor is also online. He has joined us. A number of deans that are here, some directors, academics, scientists, students that are here. We are very fortunate to have some alumni also that have joined us. Very much welcome. And quite a number of people that have joined us from around the world for this session. Let me also just recognize the efforts that Emeritus Professor Trevor Sewer, for all your efforts in making all of this possible. We really appreciate your efforts. And to welcome our guest of honor, and the partner, Carol, for traveling all the way to join us from New York. So we're very blessed to have you join us here at UCT. We host this Vice-Chancellor's Open Lectures, and this is the first one for this year to create space for anyone really who is associated with UCT or not, but have distinguished themselves in the science and their work to come and share with us their experiences, as we will do today. And tonight's guest, Professor Joachim Frank, is one such person, of course, and that is very exciting for us. He shares the 2017 Nobel Prize in Chemistry with his fellow team members, the Swiss biophysicist Jacques Dubochet and Dr. Richard Henderson of the UK, for the development of cryo-electron microscopy. Ever since I've heard about the cryo-electron microscopy, I've been talking a lot about it for the past few days. Thanks to our medical students that have made sure that we get to talk about this. Not only that, there's been a number of people around the world that have recognized the work that our Nobel Laureate has done, and there is a quote by researchers in microscopy who have recognized this work to say that the roles of cryo-EM for the advancement of life sciences, medical sciences, and drug design are immense. And it's really no exaggeration, as they said, to say that the promotion of technological development and innovation in the cryo-EM field to maximize its power is one of the most important issues for the future of human society. And therefore, it's no surprise, Jeremy, that you are also leading a unit that is trying to get us to be part of that vanguard in doing this work. And we of course recognize the importance of the infrastructure to help us move forward and really achieve what these researchers are talking about. At UCT, we have been developing the cryo-EM capability for more than 20 years now in the electron microscope unit, as I'm told. And I'm also reliably told that they operated remotely. Electron microscope operated remotely. Well, I'm hoping that we'll understand more how that's done later. But for that work that is being done here and the number of people that come from different parts of the country and parts of the continent to come and train here and do their research here, that shows us that we have an opportunity to advance this field of cryo-EM. I also had one of the students who was here and went abroad to go and do research on cryo-EM write to me say, well, the reason I am overseas is because we don't have the infrastructure that I need to do my research. But I would like to come back. But if I do come back, I would like to make sure that that infrastructure is available. It was my first week, guys. First week on duty. And then on my second week, I happened to have dinner with Jeremy and Trevor and others, and same thing. Prof, you know, it would be great to have cryo-EM here at UCT. And I was like, okay, so how much is that? The original quote that we had was 60 million, but now it's probably at about 80 million. And then I looked at Trevor, and that was Jeremy. I looked at Trevor. Trevor was like, well, round it off to 100 million, because you really need the whole ecosystem for it. So the conversation has started in this regard, and I think that in a way we can acknowledge the progress and the investments that have been made over the years, and I just want to acknowledge a number of generous investments that we've had here at UCT, from the Carnegie Corporation of New York, the Wellcome Trust, the National Research Foundation, the Sean Zuckerberg Initiative, the Science and Technology Facilities Council in the UK, and the Department of Science and Innovation at the time, now Science, Technology and Innovation, for the investments over the years. So all of this work that has been done, I think, has contributed to where we are at UCT, and the culmination of all of it has led to this point where we are hosting such an esteemed scientist who's going to share with us his experience in being part of this Nobel Laureate team that was awarded because of cryo-EM. And I think that in as much as the work that has happened here at UCT can be considered relatively young when it comes to cryo-EM, their team has trained over 31 young people in cryo-EM, and 25 of them have already achieved master's degrees. 16 of them have earned their PhDs in the subject. And they've taken up positions here locally, but some overseas, again, because of lack of opportunities in terms of the infrastructure here. At this point, UCT is the one that we know to offer the only cryo-EM facility in South Africa, and they work in close collaboration with international facilities such as the Diamond Life Source. So the opportunity to host you, Professor Frank, this evening is not just a great honor for UCT, it also is a landmark event for us and our cryo-EM staff and researchers in Africa, and our students as well. So Professor Frank is a German-born biochemist who is now American. He received a bachelor's degree in physics from the University of Freiburg in 1963. And this was interesting to me because I knew his work to be about ribosomes. And the next thing he says, no, I have a degree in physics. I'm like, how does physics and ribosomes come together. And it's like, methodology. Okay. And from 1970 to 72, as part of his postdoctoral fellowship, he worked at the Jet Propulsion Laboratory in Pasadena, California, and at the University of California, Berkeley, and in Cornell University in New York. He was a visiting scientist at the Max Planck Institute of Biochemistry in Munich from 72 to 73, and a senior research assistant at the Cavendish Laboratory from 73 to 75. But I think most importantly, and for the work that is credited for his prize in the Nobel Prize, it was when he was with the Wadsworth Center for New York State Department of Health at Albany as a senior research scientist in 1975. Beginning in 1977, he also started holding appointments at State University of New York at Albany. And in 2003, he joined Columbia University in New York as a senior lecturer, and he became a professor in the Department of Biochemistry and Molecular Biophysics, and in the Department of Biological Sciences in 2008. So, Prof, on behalf of UCT, I'd like to welcome you, and we all look forward to hearing your first-hand account of the application of cryo-EM in molecular medicine and drug design. You may take the floor. Thank you.
It's a great honor to be here. It is a great pleasure for me to be in Cape Town again. And in 2006, of course, I had no idea that such honors would be bestowed to me. So I'd like to tell you about cryo-electron microscopy, a new foundation for molecular medicine and drug design. So essentially, I'm running with the little abstract that has been distributed. So I'm just running you through the abstract very quickly, and then I'm going to talk about the subject to a general audience. I've been told that the audience is quite general, so I have to build the subject from ground up, and I hope you can understand this. So a cryogenic electron microscopy, or cryo-EM, has transformed the way we can study biological molecules, as it enables us to determine structure of molecules freely suspended in solution as single particles, and to gauge that they change their shape as they enter another. Freezing by rapidly plunging the sample into a cryogen at liquid nitrogen temperature is necessary to trap the molecules in a fixed position during imaging and to reduce the damage inflicted by the electrons in the process. And following the introduction of novel cameras for detecting single electrons in 2012, near atomic resolution has been achieved for many molecules and molecular machines of biomedical relevance, including many ion channels and receptors. And in this way, cryo-EM has evolved to become a major technique in drug design. Most recently, it has contributed in a crucial way to the development of vaccines and cures for COVID-19 and other pandemics. So the cell is essentially a chemical factory. There are many organelles that you can see here that all work together to keep, to originate life processes. And all life processes involve interactions between molecules in the cell. And a large number of these are proteins. And to understand how molecules interact in healthy or sick organisms, we need to know their atomic structure, the position and identity of each atom. And up here, is the periodic table of elements. And the most frequently elements are shown here. There are only a few: carbon, oxygen, hydrogen, phosphorus, nitrogen, and calcium. Now here is an example of a human protein, hemoglobin, which carries oxygen in our blood. About 10,000 atoms of hydrogen, carbon, nitrogen, oxygen, and sulfur, plus 4 atoms of iron. And the iron carries the oxygen. The unbelievable complexity of such a molecule, and we cannot really see how they are connected and how they are organized. We just have to know how proteins are essentially designed. So they are. I just showed you a very strange way to present it, but there is another organized way to present it as a ribbon diagram. And that shows how the atoms are bundled up and connected along helices. And the protein molecules in our cell are made by little molecular machines called ribosomes. And ribosomes string amino acids together according to instructions in the genetic blueprint, the DNA. And here's the central dogma. Now, when I started my work, the central dogma was only a few years old. You know, it's like, it was 1953, the DNA, the structure was discovered, and a few years later, the central dogma emerged, formulated by Francis Crick. And so you have to imagine that a physicist coming into that kind of context is essentially very bewildered. And it takes a lot in order to get into biology. So the central dogma, what it says is the DNA that carries all of the genetic instructions is being described at any moment by a molecule which is called RNA polymerase. And it just reads, that molecule reads the DNA and copies it. And that copy, that blueprint, is the mRNA. And the mRNA then is free to go into the cell, and then it can be translated, the code that can be translated by the ribosome. So the ribosome reads the code, translates it into a sequence of amino acids. The amino acids are the elements, and strings it together in the so-called polypeptide. And that polypeptide is really essentially a protein. The polypeptide coils together to become a functioning protein. So from that, we already know that a protein really has a linear structure. And a protein is a chain of amino acids, polypeptide, bunched together like a coiled up chain of Christmas ornaments, okay? So this is the easiest way to think about it. So there is a primary structure, the linear structure, and there's a secondary structure because the amino acids in the linear structure, they have some affinities to one another. And according to this, they have a preference to arrange themselves into alpha helices or so-called beta sheets, two-dimensional structures. And those elements then bunch together into the so-called tertiary structure. And then after these are formed, the whole protein is then formed in its final quaternary structure. In a healthy organism, all these processes go smoothly. All molecules in the cell are made correctly from the right genetic blueprints, and the immune system protects the body from hostile organisms like viruses or pathogenic bacteria. Many diseases are caused by infections. Viruses, bacteria, or parasites invade the body and cause havoc. To fight infectious diseases, we need to know how the life functions of these invaders differ from ours on the molecular level. Many diseases are caused by mutations. The genetic blueprint contains an error. causing the wrong amino acid to be put at a critical place in the protein, which causes it to malfunction and compromise the organism's health. This can also affect the immunity of the organism, its ability to defend itself against infectious diseases. So today's medicine is guided by the knowledge of the way molecules are built and the way they act in concert. With that knowledge, effective drugs can be developed, which are small molecules that are designed to bind to the target molecule and thereby change its behavior. This, in a nutshell, is the definition of molecular medicine. But how do we determine the atomic structure? That's the source of the knowledge that we need in order to conduct molecular medicine. One of the ways of determining the structure has been X-ray crystallography. The molecules can be made to form crystals. They arrange closely packed in regular order. And in that case, the atomic structure can be determined by this technique. And a very famous example, again, with hemoglobin, is Max Perutz and John Kendrew, his student. They received the Nobel Prize in 1962 in chemistry. And they were among the first to apply X-ray crystallography to proteins. X-ray crystallography has been around since 1915 or so. It has only been used to look at inorganic material. And only more recently, in the 50s, it has been applied to visualize the structure of proteins. But what can we do when the molecules don't form crystals? Could we use direct imaging of some sort, light microscope? Well, the light microscope doesn't work. The molecules are much too small, even much smaller than the wavelength of visible light. Even large molecules, such as the ribosome, for instance, would just look like a blur. And that microscope is the electron microscope. Now, we can see light microscopy and electron microscopy in strictly parallel. We have an object, which is an object, and then a glass lens. And a glass lens can be ground to perfection. And then we get an image formed, and then we can observe the image or record it. But the highest resolution is 200 nanometers, which is half of the shortest wavelength. Now, in a strict parallel to this, we can have magnetic lenses in the electron microscope. So instead of photons, we have electrons. An image is formed from the object. Now, the difference, the magnetic lens, that the electrons have a much shorter wavelength, and the resolution is better than 0.1 nanometers. There is one big difference between these differences, of course. This is a very complicated instrument, and the electrons can only move in a vacuum. The other big difference is that as I mentioned, the glass lens can be ground to perfection. This cannot be done with a magnetic lens. Only a very small part of the lens really is perfect. And outside of this, the image becomes very, very blurry. So in electron microscopy, a lot of compensation, a lot of correction is needed in order to get a perfect image. So here is the transmission scope in all its glory, but it's essentially a very old model, because the image is still recorded on a fluorescent screen as it used to be, and there would be an observer here that looks at the screen. And if you tilt the screen away, you can do the recording on a plate camera. This is no longer true. Instead, we have a camera, an electronic camera down here. So let's just go in detail through here. this is a column which is in high vacuum, because in air, electrons get stuck within a few millimeters. Up here is a high voltage source, and then an electron gun, which is a cathode. And in a very small space between cathode and anode, the electrons are accelerated to highest speed, to 100 kilovolt, 200 or 300 kilovolt. And then comes an illumination system which bundles the beam so that it hits the sample. The sample is very close or even immersed in the objective lens. The objective lens is the most important lens of the microscope. And then an image is formed in between here, and that image is then magnified by the projector lenses until a final image is formed. And what do we see now? Well, it's a transmission microscope, which means that the electrons go through the object, and we observe what arrives at the end. So the object needs to be transparent. The object has to be very thin. Now, a projection is formed. We don't really have a very good idea of what a projection is in real life. Projection is not a shadow, okay? We don't have any sharp boundaries and inside nothing and outside everything, but rather the projection is really an integral. It's like a sum of all components that the electron or the light goes through. So the best example is really a jellyfish. Okay, the jellyfish we can see from different directions, and depending on how thick material the material is, we see different intensities. So what I just told you is that we get from a three-dimensional object, two-dimensional image. How do we get back to the three-dimensional? Well, this is the so-called three-dimensional reconstruction. So we have images that are formed in the electron microscope. And a three-dimensional image can be reconstructed in the computer if a sufficient number of projections are available. And these projections have to be from different directions, okay? That's called 3D reconstruction. The tilting, by tilting the object in the microscope to collect images at many different. So the pioneering work in 3D reconstruction was done by Aaron Klug and David de Rozier. They reconstructed the bacteria phage tail using 3D reconstruction. And they make use of a very important theorem, which is Fourier theorem, that was invented or discovered in 1915 by a German mathematician, John Radon. And of course, at that time, there was no computer available and no reconstruction could be done. So the absolute pioneers in this area were Aaron Klug and David Rozier. In this case, it's a very special case because the molecules of the phage tail were arranged in a helical structure. So they were crystallized in a particular way. And so Aaron Klug and David de Rozier were able to make use of this very high symmetry, which meant that a single view already gave them the information all around the object. Obviously, we would need electron microscopy or electron tomography would mean that we have to tilt the object in all different directions. But the problem there is that you accumulate radiation damage. The electrons destroy the molecule in the process. And so working with my mentor, Walter Hoppe, at the time, I got this idea of the single particle technique. Instead of tilting the molecules, why not make use of the fact that they are randomly oriented already in the sample? So they have already tilted themselves. And all we need to do is just collect many, many molecules that sort of randomly sample the viewing directions. So single particle technique means structural information is collected from images of single, which means unattached molecules in many copies. And the molecules are free to assume all naturally occurring conformations, which is not true in the crystal. In the crystal, they're sort of packed, or they are in a straitjacket, okay? They cannot move, and they are in one fixed structure. And the molecules are randomly oriented. That's, of course, the most important premise of the technique. And a single snapshot may already give us hundreds of particle views. As we collect more snapshots, more orientations will be covered until we have enough for reconstructing the molecule in 3 dimensions. That all sounds great, right? But unfortunately, the work is only starting after this because we don't know the orientations. We know nothing. And so everything has to be done by computer programs. And my claim to fame is to develop these programs at a time when nothing was available. So the alignment of images, the sorting and classification of images, finding projection angles, reconstructing the object in 3 dimensions from random orientations that had not been done before. So all these different things were new, and I had to create a workbench of programs, something like 400 programs that all were able to work with one another. So, and we called this workbench of programs SPIDER. So SPIDER was one of the first programs at the time that could accomplish these kinds of things. The first reconstruction that was done on this principle was in 1987. It was a reconstruction of the large subunit of the bacterial ribosome. I told you about the ribosome before. Ribosomes are always composed of 2 subunits, a small subunit and a large subunit. And so the large subunit was the first structure that we were able to reconstruct and publish. And the important thing is that this was not a cryo technique. There was no cryo-electron microscopy at the time. There was electron microscopy. And we used negative stain at the time. Then came the cryo part from cryo-EM. It came from a completely different direction. It came from Jacques Dubochet, who was honored together with me in 2017. So he introduced the plunge freezing, frozen hydrated specimen preparation, and the concept of vitreous ice. Well, before Dubochet, before 1981, vitreous ice was unknown. It was unknown that water can be frozen into a form in which it is essentially glass-like. And it only happens when the temperature change is very, very fast. So if the temperature change is slow, then the water has the opportunity to form crystals. And crystals would be very bad for molecules. They would crush the molecules. So here you have the contraption, which is very simple contraption, can be hand built, but nowadays it's a very complicated apparatus. But you can essentially make it with $200 kind of equipment. So you have a rod that is held here in a particular position, and that rod carries tweezers, and at the tip of the tweezers we have a grid, and now you put a droplet of the solution that contains the molecules onto the grid, and then you very quickly blot the excess away. And that's a very delicate operation because it has to be done just right so that the water is very thin. This film of water that remains is very, very thin. This rod is released so that the grid dives into liquid ethane or propane, which is on liquid nitrogen temperature. Only when you have the very fast transition do you get vitreous ice, which is the only way to keep the molecules in a native state without crushing it. So this is how it looks when you use a commercial apparatus in which the blotting is done in a computer-operated way. This is the kind of images that you get. Well, they look like salt and pepper because you have to be very careful with the electron exposure. You only use very, very low exposure because otherwise the molecules are too much damaged. I told you about the merger of two techniques, completely different techniques. One of them is computational and the other one is sample preparation. And these two techniques merged in the late 80s. And then the single particle techniques became very successful. And these were the first molecules that we were able to look at for the first time in 3 dimensions because they had not been able to be crystallized. And that was a wonderful time with many, many different discoveries. But at the time, nobody had any idea that we would ever get to atomic resolution. We thought, well, this is a nice ancillary technique because it shows we can do experiments, we can see how molecules sort of change their domains, how they move from one state to the other. And so it was sort of ancillary to X-ray crystallography, which was sort of the serious kind of science. But then came this resolution revolution. Now here I'm really giving you a list of things that happened between these, the images that I just showed you in the 90s and later on in 2012. First of all, electron microscopes were interfaced with the computer. The introduction of automated data collection. The computers became much faster and more powerful. And then there was such a thing as 3-dimensional classification. It was possible through ingenious programs to extract multiple structures from a mixture of the sample. Now this is something completely different from other techniques or from X-ray crystallography. Because you can have multiple molecules. We can even have different molecules all in a sample. And these methods make it possible to extract them all separately. You can even see how molecules change their shape in the thermal environment. So you can have different clusters of molecules that are in the different states. And then came the huge improvement of image recording quality by the switch from film to electronic camera. So these were cameras that were able to detect single electrons. And this was a huge boost in signal-to-noise ratio and resolution. And then was the coinage of this term, resolution revolution. All of a sudden, it was possible to get to very close to atomic resolution, and now we are even at close to 1 angstroms. Now we talk about these unbelievably large and powerful instruments. Initially FEI, and FEI was taken over by Thermo Fisher. JOL has come out with a similar, a very powerful machine, but that's the kind of machine that should really be, has its place here in Cape Town, you know. And I'm gonna come back to this later on. So the pictures look like this, very crisp, much better definition than they were before. And now we are really in an era where we can go in a workflow from the sample, from where the molecules are free in solution, and then we have they're on the grid, and then they're frozen, and then we get recording of images, which is always, each image is essentially a whole stack of images. Then from then, we have an automated program that finds the molecules, and then we have two-dimensional and three-dimensional classification, 3D reconstruction of all the different forms of the molecule that we find, and then we can build an atomic model. And atomic models are now also built with programs that are guided by AI and highly automated. So here, then I give you an impression of the three-dimensional structure of the ribosome, which we got in 2016 at a resolution of 2.5 angstroms. And here, obviously, the molecule, the ribosome doesn't have color. The false color is being used here to show the different resolutions. So the very high resolution, up to 2.3 angstroms, is in the interior, and it's depicted in this deep blue color. And at the periphery, there's the molecules has more of an opportunity to wobble around, so the resolution is lower. And now, if you type in cryo-EM structures into Google, then you see pages and pages of structures, okay? It gives you an idea that this is now a widespread activity everywhere in the world. Many people use cryo-electron microscopy in fundamental research, in drug design, and so forth. So with the knowledge gained by high-resolution cryo-EM, the origin of many diseases can now be understood, and effective cures can be developed. Vaccines, antibodies, antitoxins, antibiotics, all can be designed based on the knowledge gained by cryo-EM. Small molecule drugs can be designed that stabilize the target molecule and enhance its function, suppress its function, for instance, by changing its dynamics or by blocking its interaction with a functional partner, or change or modulate its function. The development of cures for infectious or insect-borne diseases like malaria and tuberculosis require the detailed knowledge supplied by structural biology. For example, of cryo-EM advancing molecular medicine, I give 4 examples. One is the human CDK activating kinase, which offers the potential designed in fighting cancer. Cystic fibrosis, transmembrane conductance regulator, CFTR. This is a channel that malfunctions, and these are caused by mutations. And this can be targeted by drugs. Ryanodine receptor, RYR2, it is affected by many mutations which are implicated in several heart diseases, and is a drug target. COVID-19, the spike protein SARS-CoV-2 virus, of the SARS-CoV-2 virus, we can recognize, we can look at the structure and dynamics of the interaction of the spike protein with a human host, and can look at the mapping of effective antibodies. So cancer, human CDK activating kinase, is a very important molecule that controls, that is sort of very high up in controlling controlling the cell, is controlling transcription. And so it is important in the proliferation of cancer. So it is a very important drug target. And so it's involved in the cell growth and division, and regulates transcription initiation and the cell cycle. And due to its central role in cellular physiology, it has been identified as promising target for cancer therapeutics, and is a possible target for antivirals. So here is a structure which has a resolution range from 1.8 to 2.3 angstroms, and that's a range that is sufficient for definition of the drug targets. Cystic fibrosis is caused by the malfunction of a particular ion channel, and here we have a molecular structure. So cystic fibrosis is caused by the malfunction of the CFTR ion channel, which is a chloride channel. And here we have the atomic structure, which was first solved at the Rockefeller University. And here are the many missense mutations that are implicated in cystic fibrosis. You can see how many mutations there are that can possibly cause malfunctions. And it was really heartwarming to receive this message. This was a few weeks after I was notified of my Nobel Prize. Dr. Frank, I want to add to the chorus of congratulations that you have no doubt received since the Nobel was announced. I have a special reason to send a thank you as well. My 7-year-old grandson, Jackson, was diagnosed with CF as a newborn in 2010. I have attached news that we received this week regarding his 10 months on combination therapy. I wanted to share this as a small expression of my and Jackson's gratitude. This was so unbelievable to get this message. Then I'm switching over to heart diseases. This is the calcium release channel, and there are 3 versions of it. The RYR1 is implicated in skeletal diseases, and RYR2 in heart diseases. We, in our lab, got the structure of the skeletal one in 2016, close to atomic resolution. In the heart version of the channel, you see how many mutations exist throughout the channel. And they're implicated in many heart diseases. There's central core disease, central nuclear myopathy, multi-mini core disease, and so forth. And so you can imagine what it means to get the atomic structure, because now we can all of a sudden develop drugs that target all these different diseases. Then finally, COVID-19. And there's this incredible story that some of you might already know. But just 2 weeks after receiving the genome sequence of the virus from Chinese researchers, this was the beginning of 2020, the team had designed and produced samples of their stabilized spike protein. And it took 12 more days to reconstruct a 3D atomic scale map of the spike protein and submit the manuscript to Science, which expedited its peer review process. And so this study was published on March 13th, 2020, just a few weeks after the sequence arrived. And you can see it was an incredible feat, and it contributed towards the development of the mRNA vaccine, because this was the important first step to essentially reproduce the spike protein in your wet lab. And this brings me to other applications in which we now use cryo-EM to look for neutralizing antibodies against multiple epitopes of the spike protein. So antibodies can target particular sites of the spike protein and essentially render it ineffective in lodging on the human receptor. And so people continue to learn now about where these antibodies bind, and then the knowledge of this can help us design more effective antibodies as the whole disease mutates. And here is a Thermo Fisher white paper. This shows an example for high throughput cryo-EM epitope mapping. High throughput really means that these models can be built within hours. So the technology is developed to such an extent now that it can be an effective high throughput technique. So this is from the same paper where you can find out the positions of epitopes where antibodies bind. So in closing, I'd like to show you how the data bank of three-dimensional reconstructions reflects the exponential increase in research done all over the world. And this is far exceeds now the number of depositions from X-ray crystallography. So in conclusion, in recent decades, medicine has become very powerful by leveraging the knowledge of molecular structure. We speak of molecular medicine. X-ray crystallography has been the traditional technique to determine the structure of biomolecules, but many molecules do not form crystals, so the cure to many diseases was out of reach. Cryo-EM, which does not require crystals, is a new transformative technology for imaging biomolecules and determine their structure in their native states. Many biomolecules as ion channels and receptors that are hard to crystallize for X-ray crystallographic studies can now be imaged at high resolution. Based on the knowledge of the structure of biomolecules, drugs can now be developed to combat many diseases for which there was no cure. And high-end equipment makes it difficult for individual academic institutions to catch up with the cutting-edge technology. So it makes sense to pool resources into a national center. Cape Town University's structural biology facility is already superbly equipped and staffed as a national research facility, but it lacks a high-resolution electron microscope of the cryo's caliber. And given the prevalence of many infectious diseases and the persistent threat of coming pandemics, the procurement of such an instrument should be a national priority. Only in this way, academia and industry can partake in the worldwide development of cryo-EM-based molecular medicine to combat many diseases for which there have been no cure. Concluding with the image of us standing on top of our building, this was in 2018, and at that time we had little knowledge about the pandemic that was about to strike. And so we are still a very happy crowd here. And I'm holding the poster that was specially produced by the Nobel Foundation. Thank you very much for your attention.
He told me a story of a 13-year-old who is brilliant, smart, who's already doing incredible science, but because of the laws in the US that prevent old men like him from speaking to young people without getting a permit, he has to speak to this 13-year-old through the parent. Huh? 16, okay. Well, 13 and 16. And for me, I think when I heard this story, it was incredible just thinking about the multiple generations between them and the people that he's still influencing today in terms of mentorship and developing young and future scientists. Sorry. And I just want to share that with you to say that you would wonder why someone at his age would travel so long to come and spend time with you. His inspiration comes from a very deep place for what he does. You know, I was actually surprised that one of the things that triggered him from coming, into coming here was an email from some of our medical students. Even though, of course, they couldn't really do it because they need the resources to bring him, they had to reach out to Trevor to convince all of us. But it's incredible the role that the young people and also his part in supporting and encouraging young people, and I just want us to give him a round of applause for that. The last thing I would say, you will also understand why they wanted this to be a VC's open lecture. You saw in the concluding remarks, the instruction, the mandate that I was getting. And I wonder if we've got a dish here. I want to start circulating a dish to collect coins, to start donating in order to get that instrument, the cryo-EM instrument. It looks like this is the only way we are going to be able to move forward in terms of advancing cryo-EM here. But ladies and gentlemen, we have been blessed and honored the past few days, the past week, to host a legend and highly esteemed scientist in molecular medicine, someone who has contributed greatly to molecular medicine and structural biology, and we're very, very, very honored, Prof, to have had you join us and share with us your experience, your journey, your knowledge, and for encouraging our young scientists and inspiring them, and also encouraging us to, and supporting us to equip ourselves with the necessary infrastructure to continue this work. I know that a number of people are inspired to move forward. I've already received a long love letter from Trevor Sewell. with a number of requests, and I'm sure that your visit would have the necessary impact that it is meant to have. Even as you leave, you should know that you've left your footprint in this place. And as UCT, and on behalf of everyone at UCT, I want to thank you and Carol for taking the time to join us. Thank you very much. And can we give him a round of applause?